Usually when we talk about a medical diagnosis, there's this underlying expectation of mechanical precision.
Right.
Like you fall, you break your arm, the x-ray shows that jagged white line, and the attending physician just, points to the film and says, there's the problem. It's binary. It's broken or not broken.
Yeah, it's clean, the pathology is localized, and the solution is, well, it's usually structural.
Exactly. But the moment you step into the world of oncology and metabolic health, that x-ray machine effectively becomes useless. We are suddenly looking at a diagnostic landscape that is just incredibly murky.
Oh, absolutely.
The lines between cause, effect, and systemic dysfunction are blurred, which is why our mission today is to, well, clear up those muddy waters for you. We are looking at the hard clinical data from the academic textbook, The Link Between Obesity and Cancer, edited by Raman Mirzad.
It's a fantastic text.
It really is. And if you are listening to this, whether you are a medical student prepping for boards, a young healthcare professional, or just an insanely curious self-taught learner, this deep dive is specifically built for you. We are going to explore the biological and epidemiological links between excess adiposity and oncogenesis.
And we're going to try to do that without getting bogged down in, the convoluted jargon.
Right. We'll skip the jargon, but we are keeping the crucial scientific mechanisms completely intact so you can understand exactly how excess fat acts as a literal engine for cancer development.
Which is, a framing that represents a massive paradigm shift in modern medicine.
Oh, yeah.
For decades, the medical community viewed obesity primarily as an indirect risk factor. It was considered a condition that might make a surgical intervention more complicated or perhaps increase the likelihood of cardiovascular issues down the line due to lipid panels.
Right, like a secondary issue.
Yeah, exactly. But this text forces us to abandon that passive view. Obesity must be approached not merely as a risk factor, but as a chronic systemic disease in its own right. It structurally, physiologically, and functionally deranges the human body. And it acts as a direct, aggressive driver of tumor development.
To really grasp that, I think we have to challenge a very persistent misconception about human biology. Most people are taught to think of body fat as a passive storage unit, like a biological savings account for excess calories that just sits inertly under the skin or around the organs.
Right.
In reality, we need to view adipose tissue as a bustling, highly active chemical factory. It is constantly pumping out endocrine signals, hormones, and inflammatory markers.
A chemical factory that, in the context of obesity, has completely dismantled its own quality control protocols.
Oh, that's a good way to put it.
Yeah, the signaling becomes erratic, and the production lines start manufacturing compounds that actively damage the surrounding environment.
So before we pull out our microscopes and look at the cellular floor of this rogue factory, let's establish the sheer scale of the issue on a population level. Like, why is this such a critical, unavoidable topic for anyone in the medical field today?
Well, the global burden is fundamentally reshaping epidemiology. Looking at the data highlighted in the text, global adult overweight prevalence sits at roughly 39%.
Wow.
Yeah, and clinical obesity prevalence has reached 13%. That represents a staggering threefold increase since 1975.
A threefold increase in a single lifetime. That's just wild.
It is. We are seeing a profound population-wide shift in metabolic baseline.
So how does that translate to the oncology wards?
It translates directly. Upwards of 27% of worldwide cancers are now related to obesity.
27%.
Yes. And the International Agency for Research on Cancer, the IRC, they've identified 13 specific cancers with what they classify as a convincing or sufficient link to obesity.
Which cancers are we talking about here?
We are talking about major malignancies: colon, breast, endometrial, kidney, esophageal, and gallbladder cancers, among others. And the risk scales directly with the weight.
Right.
The text emphasizes a strict dose-response relationship. For example, in renal cell cancer, a mere 5 kilogram per square meter increase in a patient's BMI results in a 35% increased risk of kidney cancer for women and a 24% increase for men.
Jeez.
It is a linear, measurable threat.
Okay, let's unpack this for a second.
Right.
We know BMI is an imperfect metric.
Right.
It's something drilled into medical learners early on. A standard body mass index calculation cannot distinguish between lean muscle mass and visceral adipose tissue.
Right.
Like a professional bodybuilder and a highly sedentary individual might trigger the exact same BMI calculation on a chart.
Oh, absolutely.
So how confident can we really be in these massive population studies if the primary measurement tool researchers are using is fundamentally flawed on an individual level?
That's a great point, and the text acknowledges this exact clinical limitation. Anthropometric measures like BMI lack nuance when you are looking at a single patient in an exam room.
Right.
But the strength of the data relies on the sheer law of large numbers. We're looking at overwhelming consistency across millions of patients in multiple large-scale cohort studies spanning decades.
Oh, okay.
When you zoom out to that macro level, the statistical noise, the muscle-bound athletes, and the outliers, it just fades completely. What emerges is an undeniable linear trend of increased mortality and oncogenesis corresponding with weight gain, usually beginning in early adulthood. The volume of the data aggressively corrects for the individual imperfections of the BMI tool.
Okay, yeah, that makes a lot of sense. The sheer volume of data acts as a stabilizing force to the statistics. So if that establishes the unquestionable epidemiological reality, let's bridge the gap into the pathophysiology.
Let's do it.
How does the systemic population trend translate to a single cell mutating? Like, what is happening inside this chemical factory of excess fat that actually triggers a cancer cell to develop?
Well, the transition from excess calories to cellular mutation begins primarily with the disruption of the endocrine system. The text outlines several interconnected hormonal pathways, starting heavily with the impact of insulin.
Right, the classic metabolic hormone.
Exactly. Obesity almost inevitably leads to a state of insulin resistance in the peripheral tissues. To manage circulating blood glucose, the pancreas is forced into overdrive, pushing the body into hyperinsulinemia.
Which is a state of chronically elevated insulin.
Yes, chronically elevated.
And insulin doesn't just regulate sugar, right? It's a mitogen.
It is a potent mitogen. It actively stimulates cell division and DNA synthesis. In a hyperinsulinemic state, you're constantly bathing the body's tissues in a growth-promoting signal.
Which sounds incredibly dangerous if there's a mutated cell nearby.
It is. But the danger is compounded because high insulin levels also suppress the hepatic synthesis of specific binding proteins.
The liver protein.
Right. And this leads to a significant increase in the levels of free bioavailable insulin like growth factor 1 or IGF-1.
Here's where it gets really interesting, I think. Because IGF-1 is essentially a double-edged sword for tumor development.
Yes, it is.
It is highly mitogenic, so it's encouraging cells to rapidly multiply and divide. But it is also powerfully anti-apoptotic.
Right, preventing cell death.
Yeah, apoptosis is the body's natural, programmed cell death. It's how the body clears out damaged or mutated cells before they can cause harm. With hyperinsulinemia and elevated IGF-1, it's like obesity is putting a heavy brick on the cellular gas pedal for growth while simultaneously crawling underneath the car and cutting the brake lines by stopping apoptosis.
That visual captures the danger perfectly. The cells are being forced to proliferate and the damaged ones are being artificially kept alive.
It creates the ultimate biological environment for a tumor to take root.
It really does. And to track this clinically, the text emphasizes the utility of the HUMAR.
That's the homeostatic model assessment for insulin resistance, right?
Exactly. Rather than just looking at an A1C, OMA-IR uses fasting plasma insulin and fasting glucose concentrations to yield a highly sensitive estimate of basal insulin resistance and beta cell function.
Oh, nice.
Yeah, it serves as a vital screening tool for medical professionals, allowing them to catch this metabolic deregulation at the subclinical level long before that brick has been pressed on the gas pedal for too long.
So, the pancreas is working overtime, flooding the system with insulin. But the pancreas isn't the only organ manipulating the endocrine system here, right?
No, not at all.
The fat tissue itself starts acting as a rogue hormone producer, particularly when we look at sex hormones.
Yeah, the fat tissue essentially hijacks the body's hormone production. Adipose tissue is dense with an enzyme called aromatase.
Aromatase.
Right. And aromatase is responsible for peripheral aromatization, which is the biochemical process of converting circulating androgens like testosterone and androstenedione into estrogens.
Which becomes incredibly dangerous for postmenopausal women.
The danger spikes exponentially in that demographic. In a premenopausal state, the ovaries carefully regulate estrogen production.
Right.
But postmenopause, the ovaries shut down that production. But if a patient has significant adipose tissue, That expanded chemical factory acts as an independent, completely unregulated endocrine organ,
Just constantly churning out estrogen.
continuously churning it out via aromatase. This results in chronically high, unopposed estrogen levels circulating in the blood. Those high levels directly stimulate the proliferation of cells in the breast and the endometrium, driving the development of hormone receptor-positive cancers.
Man, it's terrifying to think about the fat tissue effectively overriding the body's natural hormonal off switches.
It is a profound systemic failure.
And that brings us to another specialized product this factory produces. We've talked about insulin and estrogen, but the fat cells also secrete their own unique proteins called adipokines.
Right.
This is where the biological mechanisms get incredibly complex.
The adipokine pathways are arguably the most direct localized drivers of the tumor microenvironment. In a healthy, metabolically flexible body, adipokines like leptin and adiponectin work in harmony. They regulate metabolism, energy expenditure, and appetite.
A nice balancing act.
Exactly. But as adiposity increases to pathological levels, the balance of these chemical messengers is severely dysregulated.
Let's break down the two main actors here. Leptin is normally our satiety hormone. Its primary job is to cross the blood-brain barrier and tell the hypothalamus, hey, we have enough energy stored, you can stop eating.
Right.
But in a state of obesity, the brain becomes resistant to that signal, so the fat tissue keeps pumping out more and more leptin to try and get the message through, leading to chronically elevated levels in the bloodstream.
And circulating leptin at those massive concentrations ceases to be just a metabolic regulator. It actually becomes a potent oncogenic signaling molecule.
Oh, wow.
Yeah, it binds to receptors on various cells and aggressively upregulates the JAK-STAT signaling pathway.
Okay, let's pause there because JAK-STAT is one of those pathways we hear about in biochemistry but often lose the context for.
Fair enough.
How does an upregulated JAK-STAT pathway actually benefit a cancer cell?
Well, the JAK-STAT pathway is fundamentally a communication line from the outside of a cell to its nucleus. When excessive leptin binds to the cell's surface, it triggers this pathway to turn on specific genes that promote cellular survival, stimulate angiogenesis.
Which is the creation of new blood vessels, right?
Exactly. And it encourages cell migration. It essentially hands the cancer cells a blueprint for survival and expansion.
Jeez. So while leptin is actively fueling the fire, what happens to the counter-regulatory adipokines? I imagine adiponectin is the protective counterbalance to leptin.
Adiponectin is profoundly protective. It functions as a powerful insulin sensitizer. It has direct anti-proliferative effects, and it actively restricts angiogenesis, basically starving potential tumors of the blood supply they need to grow beyond a microscopic size.
Okay, so we want high adiponectin.
We do, but the cruel biological irony of obesity is that as the mass of adipose tissue increases, the secretion of adiponectin inversely decreases.
Wait, it goes down.
It goes down. The protective hormone plunges, while the proliferative leptin-driven signals spike.
So what does this all mean for the localized cellular environment? We have plunging adiponectin and spiking leptin, but we also have to account for the physical space these fat cells are taking up.
Yeah, the physical changes are huge.
The adipocytes themselves are going through hyperplasia, multiplying in number, and hypertrophy swelling to abnormal sizes.
And that physical swelling is the catalyst for the next major mechanism, a state of subclinical chronic inflammation.
As the adipocytes undergo rapid hypertrophy, they literally outgrow their own capillary networks.
So the factory expands faster than the blood supply lines can keep up.
Exactly.
Which leads to hypoxia, a lack of oxygen, and eventually sections of the fat tissue start to undergo necrosis or localized cell death. And that necrosis triggers an emergency biological alarm. The immune system registers the dying fat cells as a wound or an infection, and it sends in macrophages to clean up the dead tissue.
The cleanup crew.
Right. These macrophages infiltrate the adipose tissue and begin pouring out pro-inflammatory cytokines, specifically tumor necrosis factor alpha, or TNF-alpha, and interleukin-6. The fat tissue transforms from a storage depot into this simmering, highly reactive inflammatory soup.
So we connect this to the broader oncological picture. If the fat tissue is essentially acting like an infected wound, constantly drawing the attention of macrophages and pumping out TNF-alpha, what happens to our systemic immune defenses?
Well,
Is the fat tissue creating a decoy that blinds our immune system to the actual cancer cells?
if we connect this to the bigger picture, that is precisely the danger. In a chronic, high-fat, highly inflammatory environment, the immune system becomes exhausted and deregulated.
It's just too distracted.
Exactly. The body's primary anti-tumor immune cells, The CD8 plus T cells, which are tasked with hunting down and destroying mutated cells, they become significantly less abundant and phenomenally less activated.
Wow.
They are suppressed by the sheer volume of inflammatory noise. This subclinical inflammation is the ultimate physiological bridge. It explains why systemic metabolic dysfunction inevitably leads to localized tumor progression, distant metastases, and much higher overall cancer-related mortality.
Because the immune system is entirely too suppressed by the inflamed fat to mount a defense against the malignancy.
Exactly.
It's a cascade of failures. The metabolic dysfunction plants the seed, the hormones provide the fuel, and the inflammation neutralizes the immune system.
A perfect storm.
Let's ground these mechanisms in some specific clinical realities. How does this inflammatory soup impact vulnerable areas of the body? Let's look at esophageal adenocarcinoma.
Yeah, the data on esophageal adenocarcinoma presents one of the most alarming associations in the entire text. For individuals in the highest BMI category, the relative risk of developing this specific cancer is 4.8.
4.8.
Meaning a clinically obese patient is nearly 5 times more likely to develop esophageal adenocarcinoma compared to a patient with a baseline normal BMI.
That is a massive relative risk.
Yeah.
But I have to push back here slightly or ask for clarification.
Sure.
With a relative risk that high in the esophagus specifically, is that strictly a result of the chemical mechanisms we just outlined, the leptin and the circulating inflammatory cytokines?
Wow.
Or is this an area where the physical mechanics of obesity play a larger role? Like central adiposity creates significant intra-abdominal pressure. Does the physical weight just force stomach acid upward, causing severe gastroesophageal reflux disease, which then leads to Barrett's esophagus?
That's a really great question. What's fascinating here is that it is a lethal synergistic combination of both the mechanical and the chemical. The mechanical aspect is undeniably the initial trigger. The excess visceral fat dramatically increases intra-abdominal pressure, forcing the lower esophageal sphincter open and causing chronic acid reflux.
Right.
Over years, that constant acid bathing causes the squamous cells in the lower esophagus to mutate into intestinal-like columnar cells. That adaptation is Barrett's esophagus, a well-known precancerous lesion.
But plenty of people have chronic acid reflux and even Barrett's without it inevitably progressing to adenocarcinoma.
Exactly, and that is where the systemic chemical factory takes over. The mechanical damage creates the precancerous lesion, but the underlying hyperinsulinemia, the elevated IGF-1, and that highly reactive inflammatory soup from the adipose tissue act as a systemic fertilizer.
Ah, I see.
They accelerate the malignant transformation of those vulnerable Barrett cells, pushing them past the tipping point into full-blown adenocarcinoma.
It beautifully illustrates how multifaceted the disease process is. And the text notes similar overlapping mechanisms in other systems, right? Like in colorectal cancer, the systemic deregulation of glucose and insulin acts as a direct biologic driver, rapidly increasing the likelihood of adenomatous polyps forming in the colon.
Yes. And with kidney cancer, or renal cell carcinoma, the pathogenesis relies heavily on the adipokine dysregulation and immune suppression pathways we discussed, driving a 30 to 40% association rate between excess adiposity and the malignancy.
So the biology is established, and the clinical manifestations are clear. But medicine is ultimately about intervention.
Right.
When we look at how to disrupt this chemical factory, one of the most compelling frontiers explored in the text involves an entirely different ecosystem in the body, the gut microbiome.
Oh, this part is fascinating.
We're increasingly recognizing that the trillions of bacteria in our digestive tract govern an enormous portion of our systemic health.
The text highlights that in an obese patient, the composition of the gut microflora is radically altered. You see a decrease in microbial diversity and an overgrowth of specific pathogenic bacterial strains.
Right.
This altered microbiome directly contributes to tumorigenesis. The bacteria themselves begin generating mutagenic compounds that damage host DNA, and they constantly stimulate that chronic pro-inflammatory state from inside the gut lining.
But there was one specific mechanism mentioned in the source material regarding the microbiome that absolutely blew my mind. The gut bacteria in an obese patient can actually manufacture extra estrogen.
Yes.
That entirely changes how we have to approach the intersection of gastroenterology and oncology.
The mechanism is a prime example of systemic interconnectedness. It involves a collection of gut bacteria known as the estrobolome.
The estrobolome.
Right. These specific microbes produce an enzyme called beta-glucuronidase. Normally, the liver processes excess estrogen, conjugates it, meaning it binds it to another molecule to make it water-soluble, and sends it to the gut to be excreted in waste, like a natural disposal system. Exactly. But in an obese patient with an altered microbiome, the overgrown estrobolome bacteria use that beta-glucuronidase enzyme to snip the bond. They deconjugate the estrogen in the colon, allowing it to be reabsorbed straight back into the bloodstream.
Wait, really? That is incredible. The gut bacteria are essentially rescuing estrogen from the body's trash can and throwing it back into circulation.
Yep, further driving those hormone receptor positive breast and endometrial cancers. It proves that the microbiome is functioning as an active endocrine disruptor.
It adds an entirely new layer of complexity to patient management, which, becomes even more critical when a patient already has a cancer diagnosis.
Absolute.
Obesity is strongly associated with a dramatically poorer prognosis in existing malignancies, particularly in breast, colorectal, and pancreatic cancers. The tumors residing in an obese microenvironment are highly adaptable. They undergo metabolic reprogramming, learning to employ a lipid-based metabolism.
Right. The tumor literally learns to feed off the abundant free fatty acids provided by the surrounding hypertrophic fat tissue.
The cancer utilizes the excess fat to supercharge its own growth. But the text provides a crucial, optimistic counterbalance to this.
It does.
This entire risk profile is biologically modifiable. The textbook details data showing that bariatric surgeries interventions like a Roux-en-Y gastric bypass or a sleeve gastrectomy significantly reduce not just overall mortality, but specifically cancer incidence and cancer-related mortality in morbidly obese patients.
When profound, sustained weight loss is achieved, the biological derangements rapidly begin to reverse. The fat cells shrink, resolving the hypoxia and necrosis.
The factory shuts down.
Exactly. The macrophage infiltration stops, bringing down the systemic inflammation. Insulin sensitivity is restored, dropping the mitogenic drive of IGF-1. The entire oncogenic machine powers down.
This raises an important question.
Yeah.
If profound weight loss can physically dismantle the cellular mechanisms driving cancer, how should our frontline medical professionals be approaching this?
Well, the text is definitive on this point. It requires an aggressive, multidisciplinary approach from day one. Primary care clinicians must proactively screen all adult patients and actively refer those with clinical obesity to multimodal behavioral, pharmacological, or surgical weight loss programs.
Okay.
And they must frame this intervention not simply as a way to protect the patient's joints or prevent type 2 diabetes, but as a primary frontline oncological preventative measure. Treating the metabolic disease is treating the cancer risk.
We have covered a phenomenal amount of biochemistry and epidemiology today. Let's distill everything we've unpacked into a singular clinical takeaway.
Sounds good.
The overarching lesson from Mirzad's textbook is that adipose tissue is not inert storage. It is an aggressive endocrine organ that actively drives oncogenesis through 4 overlapping mechanisms.
Right.
The mitogenic signaling of insulin and IGF-1, the peripheral aromatization of androgens into estrogens, a massive adipokine imbalance defined by skyrocketing leptin and plummeting adiponectin, and an environment of severe immune-suppressing chronic inflammation.
A comprehensive and honestly perfect summary of the cellular reality.
Thanks. Now, if you are a medical learner listening to this, here's a quick mental clinical vignette to test your grasp of the mechanisms we just covered.
A little pop quiz.
Exactly. Imagine a postmenopausal female patient presents to your clinic. Her chart indicates a high BMI and elevated fasting blood glucose levels. Based on the pathways we've explored today, what specific enzyme in her adipose tissue is directly driving her increased risk for endometrial cancer, and what crucial protective anti-angiogenic adipokine is currently deficient in her bloodstream?
So trace the biochemical pathways we discussed regarding sex hormones and the chemical factory's messengers.
Have your answer. The enzyme driving the cancer risk by converting circulating androgens into estrogen postmenopause is aromatase. And the protective, insulin-sensitizing adipokine that drops as obesity rises is adiponectin.
Excellent application of the pathophysiology. And, I want to leave you with one final provocative thought to mull over, building on that concept of tumors adapting to a lipid-based metabolism.
Okay, let's hear it.
We know that cancer cells in an obese microenvironment survive and thrive by gorging on free fatty acids. And we know this high-fat environment inherently suppresses our primary defenders, the CD8 plus T cells.
Right, it creates a protective shield for the tumor.
So if that metabolic switch is what grants the tumor survival, could the next great leap in cancer immunotherapy involve developing drugs that specifically target and block a tumor's ability to metabolize fat?
Oh, wow.
If we can starve the tumor of its lipid food source, Could we simultaneously strip away that inflammatory shield, uncloaking the cancer so the body's natural immune system can finally lock on and attack?
Wow, that completely reframes the horizon of oncological treatment. It brings us right back to our starting point. The diagnostic landscape of metabolic health and cancer might be deeply complex, and that traditional X-ray machine won't show you the problem. But once you truly understand the biochemical factory of body fat, the muddy waters finally start to clear.
It emphasizes why we must always keep questioning the underlying mechanisms.
Absolutely. Thanks for diving deep with us. Keep questioning, and we'll catch you on the next one.